So meiosis is essential to make gametes. Okay, so the gametes are the egg and sperm cells needed for sexual reproduction. The asexual reproduction, like potatoes or sugar cane or stem cuttings to propagate roses and things, they don't need to do this. Okay. In addition to that, this is also known as a reduction division, where the chromosome numbers are reduced in half. The chromosome numbers are reduced to 1/2. The parent. Okay. So the other thing is that meiosis, it generates variations. The variations are very important for survival. Okay. Say for one particular organism exists, and some disease comes in. If every organism is the same makeup, the disease can kill and wipe out that organism. And also evolution in the long run. So adaptation and evolution is very critical. Okay. So let's take a look at how the meiosis plays a role in sexual reproduction. Then we will get into the actual process of meiosis. So the sexual reproduction has two parents, a female parent and a male parent. And they are adults reaching a reproductive stage, and the female makes the egg cells and male makes the sperm cells. And this process is what they call as the meiosis. Okay. And they combine together, make the fertilized egg cell called as the zygote. Okay. So the zygote then will divide and differentiate to become the adult cells, or call it the parental cells. So you need the gametes to have half the number of chromosomes starting from the parents to make the zygote, because they are combining the amount of DNA here. Okay, so the parents are diploid with the two sets of chromosome. This is true for most of the animal cells. So the ploidy level refers to the number of sets of chromosome. I will tell you what the set means and a pair means, chromosomes. Okay. So when they divide, the gametes have only one set of chromosome called haploid, and the zygote will regain that status of the diploid because it's a 1 + 1 becomes 2. If they don't divide or reduce the chromosome number in half, imagine what happens, just like we saw in the bacterial division. 2 become 4, become 8, become 16 and 32, 64, 128. In few generations, you'll be swamping the nucleus with so much of chromosome. It is not sustainable. That's why this is an essential process that happens. Okay. So the animals, almost all animals, except one rodent in Latin America, are diploid, and some plants like cotton can be tetraploid. They have 4 sets of chromosome. 4N are octaploid, which is sugarcane, which is 8N are polyploid. That's is called many N's. Okay, so they can tolerate that, but animals cannot tolerate that. Imagine you have extra chromosome 21 in Down syndrome. It's a major problem. So it creates negative impact on that. And extra chromosome X or extra chromosome Y will create all these chromosomal abnormalities. So how does this happen is the question of meiosis. So we need to understand about the sets of the chromosome and how they divide. So the homologous chromosomes are called lookalike chromosomes. Lookalike. Okay. So these are similar, but not identical. Similar, but not identical. Okay. So what does that mean? They are also called as homologs. Okay. So you have a DNA coming from one parent, and let's call these two DNA. And you also have DNA coming from another parent. It's the same long chromosome and a short chromosome. So these two, you can call these as homolog, and you call them as pairs. Both are homologous chromosome homolog pairs. Okay, and this one set from each parent. Okay, what is a set? This is a set. Okay, and this is another set. Okay, so this is what they call as the set 2 and the set 1. Everybody understand the terminology? Because we have two, it's maybe confusing pair and a set, but if you have 23 chromosome, humans have 23 from mom, 23 from dad, make 46 chromosome. Okay, the 23 is a set, but each chromosome 1 has from maternal or paternal, so you can think about like that. Okay, everybody good so far? What the terminology, homologous chromosome, set, and the pair, the gametes, and all these ploidy level, diploid, haploid. You need to understand these terms before we get into the detail. So how the meiosis reduce the chromosome is the question, and generate variation is another question. And so I'm going to give you a high-level picture here, and we will wrap it up. So meiosis has 2 cell division, but DNA replicates once. It does not replicate twice. That is how they can reduce the chromosome number. Okay. So let's go through the stages of that. So in interphase 1, in S phase, the DNA replicates. That's where they will have this set of chromosomes. Each of them have 2 chromatids. So this is the chromatids. So after this, the meiosis 1, I'm going to give the high-level picture of meiosis 1. In prophase 1, this is the most important stage of mitosis. It consumes about 90% of the time spent on this. Crossing over occur between homologs. So the homologous chromosome, they exchange genetic material, and it results in genetic recombination. Okay. So how does that work? Like if you have the chromosome like this and the other chromosome overlapping this here, and the same happens with the little chromosome and the little chromosome like that. They overlap. The overlapping regions are swapped between the two. This is an important reason for the variation. So the nucleus is still intact when this is happening, and then only they will undergo this crossing over, and then they exchange the genetic material. Okay, so now I'm going to go to the other stages and complete it. Take another probably 5 minutes or so, we'll be done with this. So the meiosis 1 continues. You go to metaphase 1. So here you don't have prometaphase because it's 2 divisions, and they don't finally separate into prometaphase and metaphase. So in metaphase 1, the homologs line up in the middle. So when the homologs line up in the middle, if you have the cells like this, you have one homologous chromosome, another homologous chromosome, and so this can go in so many different ways. So this is how the centrioles form microtubules. So this is what the spindle fibers, they are attached to one side of the centromere, not on both sides. Attached to one side of centromere. Okay. So then what happens is in anaphase 1, the homologs separate. The sister chromatids still stay intact. Okay. And when they line up like this, So there is an independent assortment that happens here. What is that independent assortment? Is when the chromosome, say this is 1A, this is 1B, and this is 2A and 2B. So they can go in different ways. 1A and 2A can go to one cell, 2B can go to one cell, or 1A and 2B can go to one cell, and 2B and 2A can go to another cell. There's a 4 possible combinations that can happen in metaphase 1. And in humans, you have 2 to the power of 23 possible combinations happen. Okay, so the next thing that happens is telophase 1 and cytokinesis. After this is done, you have 2 cells, and each of them have only one set of chromosomes. Okay, so after they separate and they have either both of the same type, or they can have different possible combinations, 2 cells are formed with one set of chromosomes. Okay, they already becoming haploid here. Okay, one set of chromosomes. All right, let's continue the interphase 2. This is a key thing to understand: no DNA replication. So that is how it is going to reduce the number of chromosomes in half. And so here in prophase 2, this is all about meiosis 2. In prophase 2, no crossing over, but the spindle fibers attach to both sides of centromere here. So now the chromatids are going to separate of centromere, and the nuclear envelope will disassemble in metaphase 2. This is another thing to understand. So you have 2 cells, and here's the spindle fibers, and the chromatids are going to be separated. And so now you can see that. Let me put the lines here. So the chromatids line up in the middle. Okay. Chromatids line up in the middle. And in anaphase 2, the chromatids separate. That's the key thing to understand. And last but not the least, in telophase 2 and the cytokinesis 2, or you can call it telophase and cytokinesis 2, you have 4 cells are formed, and each of these cells will have one set of chromosomes and half the amount of DNA. So this is what happened in each of these cells. They have this long chromosome and a short chromosome, a long chromosome and a short chromosome. And this one has this long chromosome and a short. Okay. So the last interesting information wanted to share with you, in male, all 4 cells differentiate or develop into sperms, whereas in female, only 1 survive. That when they divide first time, 1 of them will be degraded and develop into egg cell. This is, once per menstrual cycle. And this can happen every day because only 1 in a million sperm cell will ever get to fertilize with the 1 egg cell. So, and also we need to understand the females are the one carry the baby. So in the case of animals, so they have limited number of cells that will differentiate into egg cell, and they produce once per cycle, menstrual cycle. That is called the gametogenesis that happens after the cell division is done. Okay, we are done.